Plasmodium falciparum is a protozoan parasite. It is one of 4 parasites from the genus Plasmodium known to cause the infectious and often deadly disease known as malaria. The hierarchical classification of P. falciparum proceeds as follows: Phylum Apicomplexa, Class Echinoidacea, Order Hemosporidia, Genus Plasmodium, and Species Falciparum. P. falciparum is in the phylum Apicomplexa due to the presence of an apical complex. The apical complex is a group of organelles which includes micronemes, polar rings, and rhoptries. These organelles aid the organism in host invasion. Some members of Apicomplexa have a conoid, but P. falciparum lacks this structure and is therefore part of the class Echinoidacea. P. falciparum belongs to the order Hemosporidia because it produces hemozoin. Hemozoin is a pigment made from the host hemoglobin and is characteristic of this order only. P. falciparum is in the genus Plasmodium because it uses a vertebrate host, or a human, as an intermediate host, and an invertebrate host, a mosquito, as a definitive host. It also has gamocyte, sporozoite, and merozoite stages, which are all characteristic of the genus Plasmodium. Many characteristics of P. falciparum separate it from the other plasmoid species. These species' defining characteristics include variations among the life cycle stages of Plasmodium species, including differences in morphological features, time it takes to undergo development, and the parasite's presence in circulating blood during its different life cycle stages. Within the genus Plasmodium, P. falciparum is most closely related to P. racinowi. The relationship has been established based off of the mitochondrial cytochrome B gene-based sequences. This conflicts with the small subunit rRNA gene molecular analysis of Plasmodium species, which indicates that P. falciparum is most closely related to P. gallinaceum and P. lofuriae. Despite this conflict, the first relationship is the currently accepted one. Anopheles mosquitoes are the vector for P. falciparum. The vector is the site of sexual reproduction and is thus considered to be the definitive host. Although humans undergo the most extreme side effects to P. falciparum, they act as the intermediate host to the parasite. Within humans, P. falciparum reproduces asexually. The life cycle begins when an infected female mosquito bites into a human and injects sporozoites of P. falciparum into the bloodstream. The parasites will go to the liver where they will increase their population asexually for 7 to 10 days. Within those 7 to 10 days, sporozoites will invade the hepatocytes and multiply into schizoids, which contain thousands of hepatic merozoites. Malaria-infected humans will be asymptomatic during the parasite's stay in the liver. The merozoites will emerge from the liver cells and enter the bloodstream, traveling through the heart to the capillaries of the lungs. While on their journey to the lungs, the contact with blood will initiate the erythrocytic stage by replicating within the red blood cells. When red blood cells become infected and the cell bursts, it can cause the clinical symptom of a fever. While some merozoites will continue to divide asexually, some will form gamocytes, which will circulate the bloodstream until they are ingested by a mosquito that can act as the definitive host to carry out sexual reproduction. Within the mosquito, the gamocytes will then develop into gametes, which are mature sex cells, which will develop into oocytes, which will burrow into the midgut wall of the mosquito. The oocytes then develop into sporozoites, which will travel to the salivary glands of the mosquitoes to prepare for human infection. P. falciparum has the ability to invade erythrocytes by any of at least 4 different pathways, in contrast to P. vivax, which is limited to a single receptor. The variety of options given to P. falciparum allows it to have higher levels of parasitemia than other species. In areas with low intensity rates of transmission, P. falciparum has had to adapt in order to maintain reproductive success. P. falciparum has done this by increasing gamocyte production during the beginning of host infection. By investing more into the new host transmission through reproduction, the parasite must decrease the effort spent on developmental growth. In tropical regions, P. falciparum is considered to be humanity's greatest killer. It is one of 4 Plasmodium parasites which are known to cause malaria, yet it alone causes 50% of all of the cases. Although P. falciparum is not currently considered to be a cosmopolitan parasite, it nearly was in the past. P. falciparum was once widespread in more temperate regions such as the Mediterranean, Balkans, and the United States. Fortunately, it has since been eradicated in these regions. Human impact on P. falciparum has greatly affected the parasite's distribution throughout time. From the start of the 20th century to 2002, the percentage of the global population at risk for malaria has decreased from 77% to 48%. Current research suggests a probable increase in malaria cases due to climate change, but only for specific regions such as the African highlands and the regions of Southeast Asia and South America. There is no evidence to suggest that other areas will see an increase in malaria cases due to climate change. P. falciparum has impressive genetic variation, which aids in their continuing ability to infect humans as a host. Since malaria has the ability to continuously make genetic diversity within their antigen encoding gene families, which prevents cross reactive immunity, enabling malaria to persist through the ever changing world, even with the added challenge of advancing technology. This coevolution between hosts and parasite within the parasite continually adapting is described by the Red Queen's hypothesis, which insinuates that it takes all the running possible to keep yourself in the same place. Within the last 10,000 years, malaria parasites are thought to exert the strongest known selective pressure on human genomes. One adaptation of P. falciparum is the loss of its classical non-homologous end joining, which functions to repair DNA double strand if it is broke. It was proposed that a possible selective advantage for the loss of CNHEJ in P. falciparum was to aid in mitotic recombination. P. falciparum will avoid antibody-mediated clearance by varying the antigens that they chose to express on red blood cells, extending the length of infections. That process is called antigenetic variation and is dependent on high variability. This antigenetic variation takes away the host ability to form clinical immunity. Although P. falciparum is the most prevalent and deadly form of malaria, some people are less likely than others to be affected by the parasite if they become infected. This is due to the heterozygotic advantage present in humans who are carriers of the sickle cell gene. Carriers of the sickle cell gene have one normal allele, which will cause the production of normal hemoglobin proteins, and one sickle cell allele, which will cause the production of sickle cell-shaped hemoglobin proteins. With one normal allele and one sickle cell allele, a person produces enough normal erythrocytes to be protected from anemia and enough sickle cell-shaped erythrocytes to be protected from malaria. The erythrocytes affected by the sickle cell allele protect from malaria by sickling after the parasite tries to invade it. Once the cell is sickled, macrophages through the body will carry out phagocytosis, thus distributing the schizoidogonic cycle of P. falciparum. This process allows the host body to reduce parasite replication, which keeps the parasite infection under control. Malaria poses serious risks to pregnant women and their unborn offspring. 200,000 neonatal deaths and 10,000 maternal deaths are caused by malaria every year. A recent study revealed that malaria causes up to 25% of all maternal deaths in regions where malaria is endemic. Complications of pregnancy due to malaria include miscarriage, premature births, neonatal deaths, and low birth weights. Due to the seriousness of malaria, vaccines are being worked on to end the high mortality rate associated with the disease. A study conducted in 2010 focused on proteins found in P. falciparum that could be used to create a vaccine for malaria. Myristoyl surface protein 7 is found to form a complex with MSP1, which is located on chromosome 13. If the proteins can be disrupted, then there is a possibility that its destruction would not allow P. falciparum negative effects on a human to take place.